This video is intended for those users of ChAS 2.1 to show what the new features are when upgrading to ChAS 3.0. So we're really not going to delve into the how-to and any of the real details of a single feature, but just give a brief overview of all the new features that were added to ChAS 3.0. So what stayed the same? All the features available in ChAS 2.1, so the whole genome view, the ability to view OS chip files, all those features from 2.1 are still in 3.0. We've only added new features. Nothing was removed. Chip files created in 2.1, or really any previous version of ChAS for that matter, can still be viewed in ChAS 3.0. Both ChAS 2.1 and 3.0 use a reference model file from NA33, which is HG19. But there is a slight difference. In ChAS 2.1, that NA33 reference model file was created from the framework, which was APT1. In ChAS 3.0, it's still NetEffects release or NA33 for the reference model file, but the framework itself is from APT2. And this is an update to a higher precision algorithm, which enables the development of new software features and analysis tools, such as the normal diploid normalization, which we'll talk about in a couple slides. If you would like details on the new higher precision algorithm in the APT2 framework, there is a tech note available, so please contact our support group at 888-DNA-CHIP or email support@fmetrics.com. And just in case, if you worry about which NA reference model file, NA33 reference model file you have, you will see a .r1, and those are the ones compatible with ChAS 2.1. And you'll see a .r2 in the file name, and that's compatible with 3.0. But 3.0 will not recognize the reference model file with the .r1, so there's no chance that the wrong reference model file would be selected. Hardware requirements have actually remained the same. Here's the recommended system requirements, and here's the minimum system requirements. This is available in the manual as well as a separate PDF in the release notes on our website. So the overall hardware requirements have not changed. However, there is no longer a browser-only option for the 32-bit workstations. ChAS 3.0, one of the features we'll talk about, is a database, and so it needs a little bit more power, and therefore it's only compatible with a 64-bit 8 GB of RAM workstation. What you will now see are some server requirements. So ChAS 3.0 does have this database option, and it's compatible with a Windows server, and so we do have the requirements listed here. So ChAS is comprised of sort of two modules. Right? We have the browser where we look at data and we make calls and interpretations, and then preceding that we have the analysis workflow. And this is where we take our cell files and create chip files. So we're going to start here at the analysis workflow and talk about the new features available here. So it has the same intuitive interface that we've always had, but we have some new workflows and new reports that are available within this module. So we'll talk about the normal diploid analysis. This is going to be for cancer samples run on Cytoscan. Mendelian error checking, also available for Cytoscan, and this is going to compare the genotypes between a duo or a trio of data. COMAP reporting. This is a report that you can export, which is going to look at expression experiments and copy number experiments on the same sample set so that you can quickly view for fold changes and copy number states for your genes of interest. Gene report is something quite similar. It's going to take your genes of interest and allow you to export the copy number state of those genes. You can now export genotypes out of the analysis workflow. This is something you could always do in the browser. It is still available in the browser. But if you are going to export the genotypes of many samples or even the whole genotype of one sample, it's going to be faster in the analysis workflow just due to not having to hold everything in memory, kind of like the browser does. So if you need to do some exporting of the whole chromosome or whole genome, this is a good way to go. And in ChAS 2.1, you could view OS chip files, but you still needed Oncoscan console in order to generate the OS chip files to view within ChAS. Now the analysis workflow gives you the ability to take your cell files and create OS chip files for viewing within the software. All right, let's talk a little bit in detail about a couple of these features. The normal diploid analysis. This is going to be for cancer samples that were run on Cytoscan, in which greater than 50% of the genome is aberrated, and therefore they aren't centered quite correctly. So for instance, here I have cancer sample 01 in the green data, and this was run through the normal cyto pipeline or the single sample analysis. And we can see we get no gain segment is being called here. The weighted log2 ratio is centered around 0. But when I look at the allele difference track, in a normal region, I would expect 3 tracks, and I'm not seeing 3 tracks, I'm seeing 4, which is leading me down the road of this is probably a gain. So now with the normal diploid analysis, I can run my same cell file, but instead of choosing single sample analysis, I will choose my normal diploid. And what will happen is the normal pipeline is run, but then once it gets to the end, it sees that greater than 50% of the genome is aberrated. It will go back through and now kick into this normal diploid where it looks through the file to find regions that are truly diploid, and will center the data based on those regions. And so we can see here we have a gain segment now being called. The weighted log2 is shifted like I would expect in a gain, and that matches what we're seeing in the allele difference track. So this is a great option to have if you have cancer samples that you're running on Cytoscan, and they do have some region of normal diploid. This is a great way to have a nice algorithm that will automatically center these for you. The Mendelian error check. This can be run on, again, duo or trio. Right here we're going to be looking at a trio of data, and it tells you two things really. The first one is, are the samples related? So if the samples are related, this would be child, mother, father, you will see a 1. If the samples are not related, you would get a 0. So for instance, if dad wasn't related, in this cell you would have a 0 because dad isn't related. But in this case, mom, dad, and child are related, so we get a roll validity of 1. Now down in the bottom part of the table, we get the information about the Mendelian errors. So we have our chromosomes from 1 to 22 plus X, the marker count, which is telling us all the genotypable SNPs available for that chromosome, and then the raw number of errors that were found in the trio, how many from mom, how many from dad, and then really these 3 columns are just converted to a percentage. So what we're looking for is, we expect some very low level error rate. So we're seeing right around 0.2%, let's say, error rate. But what we're looking for are chromosomes that have a higher incidence relative to the rest in the sample here. So for instance, 15, we jump to 7%. And in this case, this is telling us that on chromosome 15, there's a region, or perhaps the whole chromosome, that has a high rate of Mendelian inheritance errors. And so in this case, we see that mom has a 0% error rate, and dad also has this high 7% error rate. So in this particular case, mom, since she has 0 errors, seems to have donated both her chromosome 15s to the child, or at least a large region of them. And so we're seeing this high error rate for chromosome 15, but none of the errors coming from mom, indicating that both chromosome 15s in the child have come from mom and not from dad. We have the COMAP report. This would be for if you have run an expression experiment on, in this case, what we're looking at is breast cancer samples. So if I've run an expression experiment on breast cancer and also a copy number experiment on, or copy number samples on the same breast cancer samples, I can then combine all this data and get a table which gives me my genes of interest. In this case, I have the cancer gene list, the copy number state within that gene, and the fold change. So the fold change information is coming from our transcriptome analysis software, or TAC software, that's available on our website. You can run your expression experiment. In this case, it would be, let's say, breast tumor versus normal tissue. And I am exporting an AED file out of TAC, which holds the fold change information for each transcript ID. For transcript ID is going to be similar to the gene. It's the expression ID. And it's the Affymetrix expression ID that roughly for our purposes here in this brief description is sort of equating to the gene. So we have the gene for HER2 or ERBB2. We can see in this case that in my copy number sample, my OS chip file, I have a amplification 13, and in the expression data it's showing a fold change of 11.5. So you get other information, but this is just a brief introduction to show what you can get out of this table, that I can easily see my gene of interest, the copy number state, and the fold change from my data set. So that's what's new in the analysis workflow. Now let's move over to the other module, the browser. So the primary new features that we're going to run through are the addition of this database. So this is to help streamline interpretation. If I'm looking at my current sample of the day, it will allow me to view the segments and see if I've ever seen them before in my database. If so, what did I call them? Did I have any phenotype information with them? And it's just a way to streamline interpretation to help put annotation to my current sample. And it also gives me a place to store legacy data. To go along with this, we're now offering sample level annotations. We've always had segment level. So you could make a call on a segment, you could make an interpretation. Now we're going to do it at the chip file level, to where I could say this is a blood constitutional sample, this is an amnio, this is a breast tumor. You can customize the list to have whatever you want in it. You can also add phenotype information if you're getting that sort of detailed information about your samples, as well as now a sample level annotation. So in the end, after I've analyzed my sample, I can make an interpretation in a free text field that is housed also in the database. We're enabling some more flexible reporting options. We've always had the ability to export via PDF, but now we are allowing export in a Word format, and it just helps streamline the overall process because you can choose multiple graphs and tables at the same time. You can choose different formats, whether it be a table or this paragraph style for the segment table. Some automatic naming and some quick key exports, which I'll run through in a slide in a little bit. We've added an inheritance column to the segment table. This is going to work similar to the call column, where it's a drop-down list. And in the inheritance, you can track if it's a de novo, or if it's maternal or paternal, or any sort of custom information you would like to add in the inheritance column. We talked a little bit a few slides ago about the COMAP table, where I'm viewing my expression data and my copy number data in a table. You can also do that graphically in the browser. And then lastly, we have updated the content of the NetFX genomic annotation files. You'll now see it called NA33.1, and it just has newer content from those external resources that we can view in the detail track. We'll talk a little bit about that. All right, let's delve into this database. It is a Postgres database. It is a repository for your segment data, as well as any sort of related data, meaning any sort of annotations at the sample level and at the segment level that you've put into the chip file. What it allows you to do, this database allows you to do, is visualize what's in your database that matches or comes close to matching your current segments. And so you can do that in multiple ways. In the segments table, we'll look through and you'll see a new column. And in this column, it will tell you for your given segment, there are 7 in the database that are similar. And so what you can do then is say, okay, great, there's 7. I want to see those 7. And that will bring up a segment intersection view. And in this view, it's both graphical and tabular. So in the table, you would get information about those 7 samples that are in your database, their names, what you called that segment, their sample type. And you also get a graphical view of the way your current segment looks in relation to the segments associated with those 7 samples. And we'll show those in a couple slides. You also get a histogram view. The histogram view will be in the detail view, and it's showing the segment representation in your database. So you can see all your segments from the database in a histogram view. And we can also then filter on that histogram. So you can filter based on some of the metrics called overlap and coverage, which I talk about in detail in the Querying ChAS Database video tutorial. But basically, it's two metrics that we use to bring back segments of relative size. So if I'm looking at a 3 megabase gain in my sample, I don't want the database, when I query it, to bring back maybe whole chromosome trisomies, because they're probably not applicable. I want things of relative size. But I can adjust those to make them more stringent or lessen the criteria, depending on my needs. I can filter based on calls, just bring me back things that are pathogenic, or I can filter based on the sample type or the gender, different annotations that we put in with the samples. The database is also password protected. You have to have a certain user role in order to upload data to the database. Anybody with a user role can query the database, but you can't actually add data to the database. And then we do have an option on our website. It's a little tool called the ChAS Database Loader, or CDL. And this is for all your historic data that you may have been running in Cytoscan over the past few years, or if you have 6.0 data, or maybe 2.7M data, or Oncoscan, and you want to really bulk upload this, hundreds or if not thousands at a time. This CDL tool will allow you to do that, as opposed to having to load a handful in the browser and upload. This is just a way to really batch all your data into this ChAS database. A couple different options on how to set this database up. The first would be as a single user. So here I'm user 1, and I have a ChAS database loaded onto my laptop, and I'm the only one querying it and adding data to it. The next option is this multi-sample user. So this would be at my organization. I'm still user 1. I have colleagues, user 2, 3, and 4, and we all point our ChAS browser to this central database, and we're all adding data to it and querying from it. And so this is another way to set up. This can be on a server, or it doesn't have to be a server. It can be a workstation, a dedicated workstation that's on the domain at your organization that everybody has access to and can log into. And all you really need is the computer name in order to do that, as long as you're on the same domain. And what having these two different types of setups enables you to do is, in the manual, you will see a description of offline mode, or sometimes we'll call it airplane mode. And what that means is, here I am, I'm user 1, and I'm at my organization, and most of the time I'm in this central ChAS database when I'm querying and using the resources for that database. But I'm going to a conference, and I would really like to bring this data with me. Maybe I want to look at some cases on the airplane, or I just want to have that information with me so that when I'm listening to talks, that I could go back and see, have we seen any segments similar to what somebody might be describing in a given talk. So what I can do is make a backup of this central database, copy that little zipped backup over onto my laptop, and then use the restore function to restore it locally. And so now I can take it with me. As a general rule of thumb, you can have for every about 1,000 samples in a database, the size of the database is about a gig. So it's really not big. It's going to depend on how many segments and annotations you have for your samples, but overall it's a very conservative estimate that for every 1,000 samples it takes about a gigabyte of space. And then when I get back from my trip, I can come back and log back into the ChAS database and be back live real time again with my colleagues, user 2, 3, and 4. All right, so let's look at what some of the new graphical views look like using the database. So here we have our new columns associated with the database in my segment table. And here I'm looking at my segments loaded for this given sample. And I can see that the database is telling me you have 7 other samples that have had similar segments. And before I query that and bring that information, the detailed information back from the database, I also just want to talk a little bit about the histogram down here. We have this is the default. This is showing all the segments in my database that are either LOH, gain, or loss in this region on chromosome 14. And so I can see that, I see this quite a bit. My gain histogram is certainly a lot larger than I see in my LOH or my loss. All right, so I'm going to right click on this cell and query the ChAS database to bring back my segments. I get a new window that pops up. This is called our segment intersection window. And I get a table view as well as the graphical view. So the graph shows me my current samples segment. And you can see the breakpoints. We have these light blue lines here to help give you a view down of where the other segments in the database hit according to the breakpoints. These are all of relative size. I can see then in my table view the names of the samples in the database, as well as what I called them, and if I had any sort of interpretation that went with those particular segments. So now I've looked and, I've seen this before. The breakpoints look good to me. I can go back now and call this particular sample benign, and I will likely give it an interpretation of IGH. But you get, again, the sample type information, the phenotype information, as much information as you put in, you'll get in this table based on the samples that are in the database. Now, maybe I want to filter down a little bit further. If you get maybe 100 segments come back from 100 different samples, and you want to filter down a little bit more, maybe based on sample type, or change your search criteria, what you can do is click search again, and now we get this little window that will allow us to change the overlap and coverage metrics. So maybe I want to make it a little more stringent and move it up to, say, 70%. Or I can say, I want to make sure I don't have any cancer information. I just want blood constitutional, so I can highlight blood constitutional. And then hit OK. And my new search criteria will come back in these segment intersections with based on the different filters that I've selected in this window. And you could do something similar on the histogram side. So we talked a little bit about the default histogram, but maybe if I'm looking at a blood constitutional sample, I only want to see samples that I've marked as blood constitutional in my histogram. So I can add a histogram. You can add as many as you'd like. And I'm going to label this one blood. So I can choose how I want to view the metrics, the y-axis, and whether they have these grid lines and solid bars. I can choose if I want gain, loss, and LOH. In this case, I'm just going to add gain and loss. And then I want to highlight blood constitutional and say OK. And now here I get my new histograms, and we can see there are differences. So for instance, loss, I've had what ended up being a monosomy on this chromosome. And now when I use the blood histogram, that's gone. So this is likely one of my cancer samples that is now being filtered out because I'm not seeing it in my blood histogram. And if I'm interested in seeing what samples are in a certain histogram, I can just right click on it and say, show me the items in this region. And so if I right click, I get our segment intersection view comes back again. And I get the same table view as well as the list of segments from these samples which fall in this region of the histogram. So one of the things we've been talking about is filtering by sample type. And so how do I add the sample level annotations? So if you right click a sample and choose to edit the properties, you can edit the sample properties. You can add sample IDs. This is completely optional. You can choose the sample type. So we do offer a default list of sample types, but you can certainly customize it as well. I have added 6 or 7 different custom sample types here based on samples that I have. I can add phenotype information. I can just free text type it, or I can right click and use a snippet feature like you would previously in the when you do an interpretation of a segment. And so I have a list here from the HPO, or Human Phenotype Ontology website, but you can certainly customize this list to fit your needs. And then I can free text an interpretation about this particular sample. So for instance, I am going to say that this is a 22q11.2 deletion. And this will be saved into the database. And also, when I go to export a report, any information typed in this field here will go along with me to the report. All right, the last thing then is getting this data up into the database. So I've already annotated it. I've looked at all the segments, and I'm happy with what I have, and I want to put it into the database. So I simply right-click on the sample name and choose Publish Files to Database. You can do, I just have one loaded, but you can certainly do more. And then I get a little window here that's just sort of a reminder saying, all right, your gain is enabled and your loss is enabled because they're checked. LOH is not enabled, so it won't be published. Oh, that's right. You know what? I forgot that I had turned that off. I can hit cancel, come back here, check the LOH segment, and then republish again so that my LOH segments go up. A little bit about the sort of the maintenance side of things on the database. Based on your user role, if I'm a manager role, I can come in here and I can do a backup or a restore of a database. You can clean it up. Postgres does have automatic scripts that will re-index the database periodically, but if you wanted to do it manually, you could come in here and just click this button and that will re-index the database. If you have admin privileges, you can add users to your system. So you can give them a username, passwords, and assign their role. So again, users are going to be people that can query the database, they can do edits on an individual sample and calls, just like you could do in 2.0 and 2.1. But what they can't do is actually add data to the database or overwrite anything in the database. So you do have to be a manager in order to do that.